In today's presentation, we will explore why genetic variation is essential for evolution to take place. To understand how evolution happens, let's first understand what is genetic variation and the role it plays in making evolution possible. Genetic variation, the foundation of evolution, a prerequisite without which evolution cannot occur. Genetic variation within and between populations forms the bedrock of biodiversity, upon which evolutionary forces like natural selection, genetic drift, and gene flow act, enabling evolution to occur. This genetic variation, upon which these evolutionary forces act, is created by mutation and sexual reproduction. While mutation creates new genetic changes, sexual reproduction shuffles the existing allele within the gene pool, increasing genetic variation. A gene pool is the complete set of all genes and their various forms or alleles present in a population. It represents the total genetic diversity available for that group, which influences how the population can adapt and evolve over time. It is important to note that while genetic differences among individuals lead to variations in phenotypes, not all phenotypic variations are inherited. For instance, environmental factors, such as nutrition, sun exposure, and exercise, can influence traits like height, skin complexion, muscle growth, and overall fitness, but do not alter the genetic code passed to offspring. Think of the gene pool as a vast library of genetic possibilities. Mutations are like adding new books to the collection, while sexual reproduction shuffles the pages to create entirely new volumes. These processes ensure no two individuals are exactly alike. To summarize, genetic variation is the foundation of evolution, providing the diversity upon which evolutionary forces act. To further understand genetic variation, we will study it at two levels. 1. Within populations, variation among individuals within a single population. And 2. Between populations, variation among individuals of separate populations. First, let's explore genetic variation within a population, which can be examined at two levels: gene variability and nucleotide variability. After that, we will explore the genetic variation that exists between populations. Gene variability is measured by average heterozygosity, the percentage of loci where individuals in a population are heterozygous. Let's see how gene variability is quantified with the help of an example. Consider Drosophila melanogaster, the fruit fly, which has roughly 13,700 genes in its genome. On average, a fruit fly is heterozygous at about 1,920 of those loci, around 14%, and homozygous for the rest, resulting in an average heterozygosity of 14%. Besides gene variability, another way to assess genetic variation in a population is nucleotide variability. In nucleotide variability, variation is assessed by comparing DNA sequences between two individuals and calculating the average variation at the level of individual nucleotides. Let's understand nucleotide variability with the help of an example. The genome of Drosophila melanogaster spans roughly 180 million nucleotides. On average, two individual flies differ by about 1.8 million nucleotides, around 1%. Hence, the nucleotide variability in this fruit fly population is about 1%. 2. Now let's explore genetic variation between populations, which often arises from geographic isolation. In such isolated populations, random factors, such as mutations, introduce genetic changes. And when such isolated populations can no longer interbreed, gene flow between them is interrupted, causing them to evolve independently and develop distinct genetic differences over time. An example of isolation leading to independent evolution can be seen in finches. In the Galapagos Islands, different finch populations became geographically isolated on separate islands. Because they could not interbreed and their gene flow was interrupted, each population evolved independently over time, developing distinct beak shapes and sizes adapted to local food sources. Now that we understand the importance of genetic variation for evolution, let's explore the two primary sources that generate this diversity: mutation and sexual reproduction. In the next sections, we will delve into how mutations create new genetic changes and how sexual reproduction reshuffles alleles, both of which fuel the evolutionary process. Mutation and sexual reproduction generate genetic variation, forming the foundation of diversity upon which natural selection, genetic drift, and gene flow act to drive evolution. Mutations. Primary source of fresh genetic variation within a population? Mutations are changes in the DNA sequence that create new alleles, serving as the primary source of fresh genetic variation within a population. However, only mutations that occur in gamete-producing germ cells, within the testis and ovary, or in the gametes themselves, sperm and egg, can be passed on to offspring. Mutations in somatic cells, cells that are not involved in producing gametes, are not passed on to future generations and disappear when the individual dies. Although mutation rates are generally low in animals and plants, they tend to be higher in bacteria and viruses due to their rapid replication cycles. Point mutations alter a single nucleotide in a gene, with effects that can be neutral, harmful, or even beneficial. Neutral effects of mutations can happen in two ways. Mutations in non-coding DNA. These often have no impact on the organism because it happens in regions that do not directly code for proteins. And silent mutations in coding regions. Although these mutations occur within genes, they do not change the resulting protein due to the redundancy of the genetic code. Example of a silent mutation with a neutral effect. A mutation that changes the codon GAA to GAG in the gene that codes for hemoglobin protein causes no change in its structure. This is because both codons code for glutamic acid, leaving the amino acid sequence unchanged and resulting in no effect on the protein's function. However, some mutations have harmful effects, such as point mutations that change a protein's structure or function in ways that impair an organism's health or survival. Example of a harmful mutation, achondroplasia. A point mutation in the FGFR3 gene results in a common form of dwarfism by disrupting bone growth. On rare occasions, some mutations can be beneficial. For example, a point mutation may improve an organism's adaptation to its environment, increasing its fitness. Example of a beneficial mutation, lactose tolerance. A point mutation in the lactase gene allows lactose digestion to persist into adulthood, providing a nutritional advantage. Some mutations occur at the chromosomal level. Chromosomal mutations involve large-scale alterations in gene number or arrangement, such as deletions, insertions, translocations, and duplications. These mutations can have different impacts. Harmful effects. Most chromosomal mutations disrupt normal gene function and are detrimental to an organism's health or survival. A deletion, translocation, or duplication of chromosomes has been known to interfere with essential functions, leading to developmental abnormalities. Example: Down syndrome. An extra copy of a chromosome can disrupt normal gene dosage, leading to developmental problems and reduced fitness. Beneficial effects. Occasionally, duplicated genes can acquire new functions over time, driving genetic innovation and adaptation. This process can enhance an organism's ability to survive and evolve in changing environments. For example, amylase gene duplication. Populations with traditionally high-starch diets often have extra copies of the salivary amylase gene, AMY1. More amylase production helps break down starch more effectively, providing a nutritional advantage. Now, after having learned about mutation, in the next section, we will explore how sexual reproduction contributes to genetic diversity through independent assortment of chromosomes during gamete formation, crossing over between homologous chromosomes, and random fertilization of sperm and egg for zygote formation. Sexual reproduction reshuffles existing alleles into new combinations. Sexual reproduction generates significant genetic diversity by reshuffling existing alleles into new combinations. The three primary mechanisms facilitating this allele shuffling are crossing over, independent assortment of chromosomes, and random fertilization. Crossing over is the process where homologous chromosomes exchange segments of their genetic material during meiosis I. Independent assortment refers to the random distribution of maternal and paternal chromosomes into gametes during meiosis. And random fertilization is the process by which any sperm can fuse with any egg, resulting in a unique combination of alleles in the offspring. The cumulative effects of crossing over, independent assortment, and random fertilization ensure that each generation introduces new allele combinations, fostering the genetic variation essential for evolution. In the next presentation, we will examine the evolutionary forces, natural selection, genetic drift, and gene flow, and how they act upon genetic variation to drive evolutionary change. Join us as we dive into the next stage of evolution and uncover the forces that shape life on Earth. Don't forget to like, subscribe, and hit the bell icon to stay updated on our journey through evolution.